Welcome to the Experimental Design and Methods Breakout Session. To my right is Orit and Kristen. To my left is Holger and Guoji. We will be the breakout leaders. Okay.
You need to present yourself.
Oh, and I'm Jimmy Yi from UCSF.
Hi, I'm Marie Rosen. I'm the scientific director of the Klarman Cell Observatory at the Broad Institute.
I'm Kristen Ardley. I'm also at the Broad Institute. I have a history leading the Genotype Tissue Expression Project and working with Orit on single nucleus projects.
Holger Hein, I'm from the Spanish National Center for Genomic Analysis, and I'm leading the Standards and Technology Working Group.
I'm Goji from Zhejiang University. I'm trying to build small-scale atlases for different species.
Okay, so I think the purpose of this session is to really engage all of you in a discussion on how to best sort of design the various experiments for the Human Cell Atlas projects. And I think a few sort of considerations in terms of, we could just go down this list starting with sample sources, sample preservation and handling, and then we can discuss some of the current technical considerations and strategies, both experimental and computational, for reducing batch effects. And I think if we have time, we'll touch on new technologies and challenges.
So maybe, and only because Orit delegated me to do this, we'll start with the sample sources and discuss some of the sample preservation and handling, because we thought that was the most upfront of all of the experimental design things that we're focusing on. So we listed here, I guess we don't have it on slides, do we?
No.
Okay. Okay, we listed here sample sources as the first things to think about, and they're from living individuals, mostly in that case either blood samples from living individuals or tissue samples from surgical discards or something like that. There are also organ donors and rapid autopsy donors. Both of these last two fall into the category of deceased. And there are groups both in the UK and in the US, following on from the GTEx model, who are, and in other countries as well, who are able to collect more tissue samples from these types of donors. So I guess one of the questions is, one of the things that is worth discussing is who in the room has access to these different types of sources? What are your thoughts about them? What are the pluses and minuses? Do we think there's any sort of do we think we should propose anything systematic about collecting these? And as several of you who sat in the ethics session yesterday realized, there are different issues with collecting these individuals in terms of consents, data use, and things like that. So I guess I'll open it to anyone in the audience if you have any thoughts on these types of tissue sources or any experience with them. I think so, because there's people listening.
Yeah, there's a live stream, and I would encourage you to also introduce yourself very quickly, maybe, so that we all get to know each other.
Bruce Serrano from Cincinnati Children's. Can I just say, I think the way that you guys handled GTEx was just brilliant. The level of documentation that you had around it and the way that it's matured, it's just, I so admire that.
Thank you.
And so bringing all of that into this is, I think, tremendous. It's the way to go. In the same way that we're thinking about cell annotation, I think we need to think about sample annotation, and sourcing so getting really nice structured terminologies, ontologies, little variations of a thing type. I think we want all of that, and I think we want to have an easy to use web tool that could generate a sample table on the fly from a given collection, and could also help in people's thinking about experimental design and just setting up their study. How are they going to interact with the pathologists, the surgeons, the donors, all that stuff. So that could be really helped by a nice structured sample intake web form. And I think we could, if done in the right way, we could really get a lot of collective intelligence into these different sample types and details about the way to collect any of that, and have an interface to protocols, DIO versioning of it too. So that's one thought.
I think I couldn't agree more with you, and I think this is what links our session back to one of the other sessions ongoing, which is the Common Coordinate Framework and how to map everything, because it's very difficult with these different tissue sources. Ideally, it would be great to redo a GTEx type model for the single cell with an even deeper set of annotation, anatomical annotation rather, so that we can map on. Obviously, collecting these type of donors and tissues is very expensive to do, but to the extent possible, we're hoping we might be able to do some of that. But really, that takes us back to tying in some of these collections where people are doing disease studies or other studies of very specific tissue regions or surgical studies. How do we map those same regions in the same context? And it is a difficult challenge, and there are real benefits to doing some of these other studies on the living donors, both in terms of the freshness of the sample, a disease, if you're interested in disease versus normal. Do you want to add something?
No, I thought maybe you can add a little bit and just describe how GTEx actually sort of documented where the sample is coming from and all the metadata that's associated with, because I think that's a really nice thing to leverage in the next step.
Sure. Yes. So for those of you who aren't familiar with GTEx, we enrolled entire deceased organ donors or rapid autopsy donors. And there was a large group in the US at the NIH and people involved who created very detailed standard operating procedures for where to sample. So we sampled at 53 different tissue sites across the body. Not every site was available every time. Some organs were donated or some were considered not normal. The goal was to sample normal tissues. But we had a very detailed standard operating procedure. The sites that did the tissue collections had very rigorous training so that they would sample the same site over and over again. There is human error in this, there is variability, but every sample that was collected then had a pathology image done and pathology review, and the pathologists would comment if the wrong area was sampled or it was grossly different than something else. So there's a huge amount of metadata associated with that, which is very difficult to get on some other studies, but can be gotten on this type of study. And so it's a very rich way of trying to map back these type of details.
Having standardization on the imaging, just as the documentation. Maybe you just repeat it.
Having standardization on the imaging and documentation is really valuable, yeah.
So
Yes.
I'm Kirsten Meyer. I'm from the Sanger Institute, and we work a lot with organ donations from the Cambridge CBTM, Cambridge Bioresource for Translational Medicine. And I couldn't agree more with all of you guys that the annotation is incredibly difficult. But for example, most of our donors and samples are being taken in live operating theatres. So the time frame people have for collecting the samples is incredibly short. So there was beautiful talks in the lung on, how you can do CT scans of the whole organ and then see where you're sampling. Well, there's no way we can bring a CT into a live operating theatre. So I think as we are trying to get nice, fresh samples and also from different cohorts, we also have to accept that the practicalities are often just such that we cannot do everything.
I couldn't agree with you more, and I looked at that slide yesterday and I wondered how you went from the left-hand side with the CT scan to the right-hand side with the tissue and still had viable RNA. But it's a great challenge, and it's probably, as Rahul said yesterday, something we'll have to piece together from these different data modalities because it is too hard to do everything. And a lot of our GTEx donors were also in the OR as well, and you just can't do that. You can't take a tissue out and get everything you need. So...
But in the GTEx, you were working on SNAP frozen material, right? So in the ideal case for a single cell application, you would start with a viable live cells. So the GTEx model might not apply completely, or in the in a perfect case scenario for the HCA, right?
That's true. I mean, to get 53 tissues from an individual, we had to preserve the tissues very rapidly. So that gets us sort of into the next area we were talking about, preservation methods for the different data modalities that we want and need. So getting the fresh tissues is done more from these living individuals than it is from those. You could do it for 1 or 2 tissues, but it's really hard to do it for more than 1 from those type of donors. You have...
And that's why we actually developed our toolbox to actually be able to look at a large number of tissues that are frozen quickly. So I think in that sense, it's very useful. But I do think it's important also at the same time to try to get samples from living individuals, because that way you have all this, the full swath of tissues. I mean, from living individuals, supposedly it's, you know, they're not as stressed maybe. So it would be nice to compare and see what the differences are between the 3 different modalities of rapid autopsy, organ donors, and live individuals.
Goji, do you want to comment a little bit on sort of how you were able to get 50 tissues in China and get those sequenced, and what that process was?
So I think we have 3 sources of donations. One is deceased organ donation, and then the abortion, tissues from abortion, and then samples from surgery. The most abundant materials are usually sampled from surgery. However, you know, it's not like completely healthy, right? I think for the deceased organ donation, the issue is it usually takes longer time to process the sample, and most of the samples has a pretty bad quality. I think a good way to solve this problem is actually people should start to try J-Sandra's like split pool protocols, which utilize fixed sample for single cell analysis. I think that can be pretty helpful to get, you know, better quality data. Yep.
I'm not sure exactly which protocol you're talking about. What is it?
No, it's a fixed protocol. First you fix the cell, and then you do barcode inside the cell.
Fast frozen.
Yeah, you can do fast frozen. You can do fixation as well. Yeah, something like that. Yeah, I mean, most people don't get it working, but actually works quite nicely in my lab. That I don't know, but based on our current tests, it works pretty well. But the gene number is lower, but I think there is lots of rooms to optimize.
Hi. Morning. Yeah, just to follow up on what Dorit mentioned. My name is Asaf. I'm the Associate Director of the Center for Cancer Precision Medicine at the Dana-Farber. And we work quite tightly with Aviv, Rit, and their teams for many years now. So we basically, I was wondering if other people establish pipeline like we did together. So we have 3 main avenues. One is fresh tissue, the other is frozen, and the third one, relatively new, is frozen/fixed for spatial analysis. What we do basically is we have 4 teams that responsible to run the sample from, or even before the patient was admitted, all the way down to sequencing and analysis. We start with the operational team that's responsible to find the patients, check the IRBs, make sure that all the operational procedures will fit the scientific question, whether it's a clinical trial or investigator-initiated study. Then we have an email invite with all the allocations listed after we review that, together with the KCO team. So now with this invite, the pathology team can approach the patient, whether it's an IR or OR case, and collect the tissue as fast as they can. And we do emphasize the viability of cells and the authenticity of RNA. So we want to speed up those processes. And the next team will be the lab team, the innovation lab team. Sorry. They will process the fresh tissue and we'll move it to Orit and her team for library preparation after 10x and then sequencing. And then we'll have a roundtable with the computational biologists, surgeon, pathologists, just to integrate the data all together. So this is our pipeline, and I wonder if you have other experience similar to that. I just want to mention that in our hospitals, so Dana-Farber and Brigham and Women's, they really like the leadership of both hospitals, really like this pipeline that started with the need for fresh tissue for single-cell RNA sequencing. And now our center is responsible to collect all the biopsies for research from Brigham and Women's. So we're talking about more than 10,000 cores per year. And that's a token for the work for single-cell RNA sequencing. Thank you.
So how do you get funding for cross-study support like that?
Either it's part of a clinical trial or grants or big projects like this, or Human Tumor Atlas, and so on.
We always talk that we process a sample for live cell processing, that we process samples as fast as possible. Did you set like a limit, a minimum time that a sample needs to be transported and before the time before being in the 10x?
Yeah, we try to make everything as fast as we can. It's not ideal, it's never perfect, but I would say that if we'll keep the 2 hours framework, time frame, we'll be happy. Sometimes it takes shorter, sometimes it takes more time. Sometimes it depends on the traffic between Boston and Cambridge. The day of the marathon was really challenging.
There's drones for that now.
But I do have to say that we have these beautiful drawings of all the pipelines that we have with the time frames. So I know that with the SAF, for example, we went on a tour for the Farber. So they have like a specific place where the person from the OR and IR meets the technician, then goes to Broad. So everything is sort of calculated on like to really minimize the time to get the shortest path available, and we try our best. So I think that's.
So
and
actually,
we also process samples at if it's fresh samples, at various times of the night and day and early morning. So you need to have a team that's sort of ready to go, because, for example, we do some rare cancer types, pediatric rare tumor types, and for that we're always like, and for any other samples, we're always ready to go. And people are really dedicated and you know, they're excited to work on this and to do good. So I think that's you have to have a sort of infrastructure and a good team to do that.
But
Let's take your comment because you're there.
actually, it's a related question. So my name is Jay Shin from RIKEN, Japan. I guess Orit had presented this concept of SWAT Team, which is now part of the Human Cell Atlas. And I guess the design of that is to help train such logistics, not just abroad, but across the country. And my question is, how expendable is this to international setting, and is there a way for SWAT team to either come to different countries or some different visitors from around the world visit the SWAT team to get trained? Because I think this is one of the important questions for data standardization and sample standardization within the Human Cell Atlas. So YouTube videos?
Yeah, so I think we have several models. One is the SWAT teams that we just sent out. And we have specific funding for those SWAT team visits. So you do need funding, right? Because you need to send the people to a place and they need to have flight, hotel, and stuff, so forth. But I think these visits have been extremely useful for both our team and for the teams that we sent over. So, for example, with St. Jude, and Asaf can comment more because he was at St. Jude. We did really, we have an interest in one of the group that we went to, we have interest in the tumor that we're working on, but so that sort of also added a layer. But the nice thing was that we really did hands-on, side-by-side comparison. So we did processing of fresh tissue, and then we also froze that tissue and did processing of the frozen tissue, and then looked at the data together. So I think that was useful. St. Jude's is a very organized hospital, so they have like an amazing system of how to get the samples and how to get them quickly. So we learned quite a few things from them. They have really, really good systems in place, so that helped us. We also learned some of their dissociation methods. So, each team has their own trick. I think we had another visit, which people are just starting up with single-cell genomics. So that was, I think, a real useful experience for them. We sent all our experts over. And when I say all our experts, it means somebody who knows how to dissociate the tumor, somebody who knows how to do nuclei, someone who knows how to run the 10x, and also somebody who's a computational person, and someone who's an expert in the hospital pipelines. So I think that's one model, and I think it works really well because you really need to be in the place where this happens in order to be able to make it work. You need to be in the environment. So we have that sort of model. We also had a group from India. I don't know if they're here, but they came to the Broad, and we talked about what their infrastructure is, what are the possibilities. They stayed for 2 weeks at the Broad, and they learned sort of our pipelines. And then they had to go back, and now they have the ability actually to process tumors in India. But when we talked to them, we had to calculate how long it's going to take actually from the hospital to the lab. And we thought, it's actually better to have a centrifuge at the hospital, things like that. These are small details, but I think it always is really good to go to the place and see what the infrastructure is there. I think you have a better chance of understanding. And the other really nice thing is to have sort of workshops. So recently there was a SlideSeq workshop at the Broad. So people from the Broad came, and Faye and Evan and Bob sort of showed people how to do SlideSeq together. So there are a few different options.
So in the interest of time, and I think we are starting to get into the sample preservation, the handling component of this discussion anyway. So for those, especially, I guess, the Broad representatives who've had a lot of experience handling samples, what do you think of all of the items that we've listed here? Sort of sample transport logistics, digestion time, temperature, separation type, generating workflows, and standardizing materials across methods. What do you think are some of the most important considerations for maybe other sites that are thinking about setting up this kind of pipeline?
Could I just add one bullet point to that list, particularly for maybe where you have your NukeSeq toolbox.
Mm-hmm.
It's I'm worried about the optimizing the multi-sample type from a given collection that's suitable for spatial, for NukeSeq, for cell suspension, for other maybe other kinds of immunofluorescence type stuff. That so getting the most number of options available from a given collection and keeping that organized then preserved in the best way. Mm-hmm. Is that fair to add into that list, or?
Yeah, so I think in terms of workflows, and Asaf can comment more on this, we've been trying to figure out what is the best way to process the samples. So we don't want to just do it in one way. So, we try to do fresh when we can. At the same time, we make sure to take a piece, just if it depends, of course, on the size, right? And how many cores you have and the size of your section, what you can actually do. But we do try to have a piece to have at least one piece of the tissue frozen, for example, for whole exome and for bulk RNA sequencing, one piece frozen in OCT if we can, because OCT sort of tends to keep the structure in a better way, and one sample for FFP. We also try to just archive samples. So, we don't know what the next best method is going to be and what kind of sample preparation will be needed for that. So I think archiving is a very important aspect. We do try to write down these workflows. So we sit together, all the teams, and actually we draw it nicely, but these drawings have like a real infrastructure behind them, which we work pretty hard to preserve and make sure that everything is done quickly. So we have the pathologist meet the person who gets the sample in the IR, for example, and they put it in FFP and in OCT, and just try to do it the best we can. And to allow all assays, both spatial and single cell and single nucleus RNA sequencing, and whatever comes next.
Much like DMSO buffers too in any of it, or is that first secondary step? So, right, we're like for 10x, we're in cell suspension. Our general approach to distant collaborators is getting people to make suspensions into DMSO media and then freeze that and then send us that.
So I think that might work well for immune cells, but not necessarily in our hands for all cells. No, we don't do that. But I'm happy to hear what other people do and what kind of preservatives. I know Holger and Kristen talked about some of these things.
So we do very similar to a read that you take, once you have your sample, you take a frozen one and then the other materials used for single-cell RNA-seq. But we do keep any, like once you've got a single-cell suspension, you don't usually need all of it for your 10x, and we always do freeze those in DMSO. And actually, especially for the fetal liver project, that has been an absolute lifesaver. So once we identified a new population, we could then actually go back to that same individual and fact sort the cells and say, look, it's not just at the RNA level. We can actually show that this is a unique and distinct population. So don't ever throw anything away. Just keep everything if you can.
So we tested cryopreservation in many tissues, in many contexts. It works very well for immune cells. Immune cells from tissues are working pretty well. We had some problems with specific tumor types. So mesothelioma were very hard to freeze. Overall, I think it's a viable solution. Yeah.
Can you just talk about your best, what's optimized?
So we work on DMSO solutions, as you said. So it's
what about that upfront release of cells from these highly dense collagen tumor types? Pancreas cancer and mesothelioma.
Yeah, no, we usually we mince the tissue and then cryopreserve it, and then isolate the cells after defreezing it. I think the most important part is the defreezing step, where you gentle diffuse out the DMSO from the tissue. Once you add your medium right away, basically the cells explode. The defreezing is the most important step, also for blood cells. So as I said, for some tumors this works well, for others it doesn't at all. So I think it's very important to do like kind of benchmarking up front whenever you start with any tissue. So I don't have a general solution that works for some tissues or others.
It seems to me that a lot of us do our work out a certain protocol. Ideally, as Arit says, we try to collect as many things in as many ways as possible. And usually when she tells me we need to collect X, Y, Z, and A, B, C, D, E, F, I look at her like she's got 10 heads. But I don't think most of these protocols are out there. And so I think for the purposes of the HCA and the community, it would be really good for those of us who've done these, or any of us who collect our tissues in certain ways, to really provide those as a set of protocols. The other thing which is getting us into these best preservation and storage for different techniques is that for each new technique that comes along, most people don't try all types of materials and resources. They try one, they'll optimize it on that, and then they'll say, this is how we work on fixed tissues, or we work on OCT tissues. And it is, for those of us who have largish pieces of tissues and are able to preserve in multiple different ways, that's fine. But for most people, that's not. You can't physically take your sample, if it's a teeny biopsy from a surgical donor, and and have it preserved in every possible way. So I think, and maybe it's the mandate of the Technologies Working Group to try to collect a set of multi-frozen tissues as a resource that people can try on, and we can come up with experiments people would like to make these technologies and see how well they perform on the different types, but also to make sure that we collate and collect these upfront protocols so that they're available for people to follow exactly, because people do very different things, even talking about putting tissues in DMSO, whether you mince first or do this or do that, and we each have our own unique ways. So I don't know if anyone has any comments about that.
Can we maybe have a show of hands who's deposited a tissue digestion preservation protocol into protocols.io? Okay, so I count like 4 or 5, right? And does everybody know what protocols.io is? Anybody who doesn't? Okay, so everybody knows. Okay, I think that's helpful. And
But I've never looked at it.
well, but Kristen, maybe a question for you is for GTEx, how's that information disseminated?
Right now, all of our information and the SOPs are available on the NCI's BBRB website, and we rehost them all on the GTEx portal. But yes, putting those into protocols.io again would be another thing to do, and I guess to look there and see how many of these protocols are actually there.
So I think for protocols.io, it's nice to put and share with the community. I think this is really essential, but we also use it in the lab as our sort of notebook, right? When we get the tissue, we actually just document everything on protocols.io. So we have it's like a lab notebook. So this is really useful because then we can share it with other people in the lab. And let's say the data comes back and there was some problem with the sample, we can go back to that. We share the protocols also internally so people know which ones to use. There are a lot of comments back and forth. And now we actually spent a long time trying to write the protocols in a way that they will be the most comprehensive for people who need to repeat them. So we did a lot of really brainstorming between ourselves how to write them in a good way. So a person who's not in the lab like me can really go through it and understand exactly how to do stuff.
So let me just clarify. You're saying that at least internally you're using protocols.io as a form of electronic notebook. Is that? So I think
maybe you want to comment more.
So to begin with, we contemplated between electronic lab note, protocol.io, and surprisingly, we used a lot Slack. So Slack, I don't know if you guys know this application for delivering quick messages between groups. And as I described before, that we work with 4 groups at the Faber and then Rit and Viv's group. So we need to communicate a lot, and we need to document. And we start to document from the moment that we're being called to the pathology. So our pathology team, they look like journalists, actually. They have their iPads, and they have pockets full of stuff. Anyway, they come and they take pictures of the samples just to orient later all the assays that these samples will be allocated to. Then everything goes on Slack. And that's very useful because you have the picture, you have the timestamp, so you know how everything happened on a time scale. Then what we did is that we had the protocols.io internally, and we went back and forth, just like Rit mentioned. We integrated the pictures of the same samples that we worked on. We discussed whether that's the right thing to do, and again, roundtable discussions with as many people as you can get. And then once the protocol was optimized and once we're feeling comfortable to share it, we will share it with the rest of the team, not completely with the entire project, but we will have a full exposure still internally, and then we can go to the next layer and share it with the entire project.
Hey, Joseph Powell, Garvin Institute in Sydney. I feel like we there's been a number of tissues or sample types where there's been the right level of comparison of different tissue prep methods. We have a pretty well-established understanding of what would be the gold standard for at least a certain type of tissue in a certain disease or healthy condition. So to what extent do we need to, for those situations, essentially have a version-controlled lockdown 1.0, which is clearly communicated to the community? So if you're going to contribute tissues that particular type, right, you use that protocol. And then a list of the protocols where we have not established that level of gold standard comparison. Because I feel like we're involved with a lot of cancer work as well, and we similarly are going through the same kind of comparisons and tests. We don't really know whether necessarily they're going to be, we've evaluated them completely, or if indeed, once you've locked that down, whether it is going to be standardized against what someone else is doing at a different institute, because they are going to have substantial differences if they're not federated properly. Thanks.
Holger, do you want to comment on that since you're part of the STWG? Is there a charge to the STWG to collate all of the various protocols and start putting together a sort of version control system for them?
We started discussing like a kind of quality control system for data sets and then feeding that back to the protocols used. So kind of look at the data sets to see what bias we see, if it's a good quality, bad quality, not so good quality data sets, and then kind of feed that back into protocols.io as kind of verification if a protocol works or not. Are we discussing that? But there was no general agreement if this should work or not, because in the end, if you have a variety of protocols, you also get a variety of different cell types isolate from tissues, so there's probably not black and white solution. And having tissues prepared with different protocols probably would give you like a more complete picture overall what tissue types you have in your, what cell types you have in your tissues.
Maybe I can comment again briefly. So we did try at some stage to use the same protocol for lots of different tissues, so that if, for example, you get immune cells out, they've at least seen the same type of enzyme in the dissociation protocol. But actually, they're just so different. And at the end of the day, we kind of given up on it and said, look, we just have to use different protocols for different tissues.
Can you give an example?
So, for example, we were comparing, if something as obvious as spleen and esophagus. So if you treat spleen with liberase, the cells come out very easily. If you treat esophagus with liberase, not very much happens. So you then do a secondary step with trypsin, and then lots of cells come out. But, if you just did, the first step, really, you'd be missing lots of cells.
And then how much, let's say you took the spleen through the trypsin version of the protocol, with that, how much do you lose?
Well, actually, by the time you have, we've done it one enzyme and then the other one, and by the time you've done the liberase, the enzyme, the tissue was dissolved. There was no...
No, I understand that, but I'm just trying to, if you're trying to do comparison between the immune cells of the esophagus and spleen, you need to have them go through the same protocol. So use the worst protocol maybe on the spleen just to make sure you've got some comparability.
Yes, like with a lot of these, as I said, one of the problem was that we had the spleen tissue and it had there wasn't anything left after, we filter it, and it had gone all through the filter. So there wasn't anything left to do with the trypsin, and we didn't do this 4 different times, right? Because of cost and so forth. But I really like Holger's suggestion of feeding results from publications back into protocols.io, because some people might be really interested in epithelial cells or really interested in immune cells. So if you know which protocol works best for what cell type, that would be a really useful thing to know. And I think currently that's really not captured really well.
Because some people, as you said, might be interested particularly in the immune cells, and there's one protocol that works great for immune cells and another one that works great for epithelial cells and another one that you can see both of them. So I think that's a really important question, which we really started to discuss last night at dinner when we were thinking about this. Are there any questions from the live stream? No. Everyone's asleep. I don't know what the time is actually.
Depends on where they are.
Yeah.
Hello, 17 people online. So just to clarify, I wasn't suggesting that you need to have one protocol for a given tissue type, but we similarly use completely different protocols if we're doing a skin digestion interested in keratinocytes or APCs. But but for those of the members of the community that are not actively working on different digestion steps, but want to generate data, having a sort of a pretty clear answer, right? Well, this is considered to be the variety of different protocols which we would be, 1.0 for digesting, and what biases that might give in cell populations. Clearly communicating that, through the rest of the consortium would be pretty valuable, rather than having to sort of reinvent the wheel, each step across across a new tissue type.
Right. Maybe I should also share some of our experience. So because all of our data in that landscape are generated using single cell, so we spend lots of time in studying how to digest different tissues and combination of coagulases. And we have done lots of tests in mouse first. And then we try to preserve as many cells as possible when we get a tissue. But the process can be very long. Like sometimes if we know there is a sample in the middle of the night, then we all wake up and process the sample and do not sleep. So that's why I feel like it is too much for processing real single cell, whereas it might be much easier to get single nucleus from fixed samples and snap frozen samples. Because in the long run, we want to compare different data sets from all around the world, right? It should be a more user-friendly protocol.
So I will comment that we do have a toolbox for single nucleus RNA sequencing, which works pretty well across different tissues. You still need to test your tissue with the toolbox and see what looks best, but I think that would work very well. And then you can just, you can freeze the sample, and then whenever you want to process, then you can process also several samples together at the same time. Maybe we want to talk about hashing at some point and reducing, maybe Jimmy wants to comment on hashing and reducing biases.
I'm going to wear my moderator hat first and just summarize sort of what I heard is that the protocols are tissue specific, brought up by the UK group, and then Holger mentioned that they're also cell type specific. And I think some of us are touching on this other idea, which is that it's also assay specific, depending on whether or not you want to do single cell RNA-seq or single nucleus RNA-seq. And maybe this is a good time to then move us to the next section, which is to talk about current technical considerations. And maybe we should just start there, thinking about single nucleus RNA sequencing versus single cell RNA sequencing. There might be some benefits for the nucleus workflow in terms of standardization of protocols, but maybe we'll just open this up to the rest of you in terms of your experience with the various protocols, concerns, etc. And I can talk about hashing.
I think there's very few data out really comparing single nuclear and full cell sequencing. Did you ever do that for your tissues?
Well,
we assume that it's less data, but we don't know how much less data. And if it's really true, right?
We are comparing now. So the problem is sometimes you get very small samples and you have to choose what you want to do, and you can't do both fresh and save a piece. So it's a little bit more difficult. But we do now have a few data sets which directly compare. But again, you do need to remember that when you take a tumor and you divide it into two, there's still heterogeneity. So it's not like an exact, But it looks overall pretty good. Each method, of course, has its own biases. So
but there's less data in single nuclei. And
there's less RNA in single nuclei, right?
And you have a less complex transcriptome, or is your library as complex as a full cell? And
it's less complex, so that's one of the minuses. But still, you pretty much see the same transcriptional. You can see the same cell types.
Well, those are two separate, completely opposite statements. So either you can see subclass structure in the nuc-seq, and it's rich per that cell type subtype, meaning a good number of genes per R specific for that.
You still see a good number of genes. It's just not as high as in cells.
Okay, so can you just say something about the range of discrepancies, and when are you seeing the highest discrepancy between whole cell versus nuc-seq, and what guidelines for read depth and technology per nuc-seq?
So what I will say is that there are some cells that you actually cannot get any information for when you do single cell RNA sequencing. So for example, in our hands, adipocytes, like large cells, adipocytes, which are fatty cells, neurons, things like that, you're not likely to get when you do single cell RNA sequencing, and that's why we actually developed the single nucleus RNA sequencing. So that's kind of a trade-off, I think. We are in the process of comparing these two methods, so once we have that in a better shape, I can tell you a little bit more.
I'm specifically worried about the case. I understand, appreciate, absolutely love NucSeq for all of those reasons that you mentioned. I'm specifically worried, though, about the case where the NucSeq signal is, you can tell it's this cell, but it's nowhere close to what's happening in the picture of that cell based on the whole set, the intact cell transcriptome.
Yeah, from our data, it looks like you can recapitulate pretty well the cell states and the cell types, so I'm not really particularly worried about that.
And maybe that's where adding in some spatial methods might bridge.
So at the end of the day, if you want to capture all the cell types, right, you need to do both bulk RNA sequencing, single cell, single nucleus, and spatial. I think that would give you, and that's what we're pretty much trying to do, and that will give you the sort of, we hope, a more entire picture.
I see some new faces. Please introduce yourselves.
Ellis DeForest from the Harry Perkins Institute in WA, Australia. Now, what I was going to say is, so we've been doing a benchmarking experiment on mouse kidney. Now, the advantage with that is you've got 2 kidneys from each mouse. It's not a tumour, so if you cut a tumour in half, you could have half of it, it's epithelial cells, the other half has got all the macrophages in it. So you can't actually do a very well-controlled experiment unless it's a homogeneous sample. So we can take a kidney, we can do a cold dissociation, we can do a hot dissociation, we can do single nuclei isolation, we can do bulk RNA-seq before dissociation, bulk RNA-seq after dissociation, we can do cryostorage, methanol fixation, or fresh. And in all of these, we see different biases. And actually having a ground truth is really, really important. So I know with the warm dissociation methods, we see cell-specific stress responses. So there's some particular cells that are completely stressing out by going through a warm dissociation. Cold dissociation looks really, really nice. There's one cell type in kidney, Lupf-Henle cells, that don't particularly like it for some reason. When we do the cryopreservation, surprisingly, one of the populations is just gone. When we bring it back up, we don't see it anymore. The proximal tubule cells, which is the major cell type in the sample. And then lastly, when we do the methanol fixation, that actually looks very, very close to what we saw with the fresh. I don't know many people who have actually tried it, but it seems to work.
Without protease?
So you do cold protease, you get a single cell, or cold or warm protease, you get a single cell suspension, and then you fix that single cell suspension in methanol. So it seems to be better than what we recommended to all our collaborators to do cryopreservation with DMSO.
Wow.
So I'm wondering if we could charge you to do this for all the other tissues as well and report back to the Standards and Technology Working Group.
When are you going to get it out there?
Hopefully this year. We got some data off the sequencer just the week before we came here, but we're working on it. Has anyone tried the cold protease method?
Yeah, that's almost all we use. That Potter at my place.
Is it
my orders after we didn't have type 1 cells represented at all in our cell suspension?
Is it incompatible with site-seq? Do you strip the antigens?
I think it's not very. Nobody's too excited about that.
Okay, just wanted to check. Thanks.
Hi, Michaela Smith from Imperial College. So you guys have been talking a lot about cancer, but there's other tissues like the heart. So if you work with the heart, there's no way you can do a single cell suspension and get all the cells because the cardiomyocytes are mostly gone. So it's just to reiterate the fact that you need complementary approaches to cover all. We need to do nuclei to make sure that we capture the cardiomyocytes, and then to capture other cell types, we need single cells. So I think that it's going to be really hard to have, even for the same tissue, one protocol only, and nuclei won't be able to cover everything. For example, we see much less endothelial cells when we do nuclei preps, and when we do our cells preps, we cover much more endothelial cells.
Yeah.
So I think to cover all the cell types, we need to probably apply, as Rick was saying and others were saying, we need different
Could you
techniques.
just give a little more detail on that? What exactly is the difference that's helping the endothelial cells?
Endothelial cells, when we do single nuclei-seq, we capture very well nice heterogeneities of cardiomyocytes and most of the other cells. We still pick up endothelial cells, but we don't pick up the proportion of endothelial cells that you expect. And then when you do single cell, of course, you lose the cardiomyocytes, but you'll capture nice heterogeneities of endothelial cells. So we need both.
Yeah. So I really
Yeah.
think we need a complex toolbox and not necessarily
Yeah.
just choose
And
one
then
thing
spatial
and then expect
and,
to have everything. So I think
We'll bring
spatial
everything
and-
together,
yeah.
like single, the validation part,
Mm-hmm.
then will really tell you if you miss something or
Yeah.
what is the big picture.
Totally agree.
So we had some single nuclear sequencing in this benchmarking paper as well, and so compared to the other techniques, it was performed. It gave pretty good data, but when we correlated data set, it did not correlate at all with the others. So it type of single nuclear sequencing gives you good data, but a different type of data, because in the end you're sampling from a different RNA pool.
There are now new computational methods that are out there that try to put these types of data together. I don't know if you tried those, but there's Rahul's and Peter Karachenko and Evan McCosko. They have like all these like new and nice methods, which hopefully will help look at this data.
Through computational correction, you can do a lot, but in the best case, you avoid that upfront.
Yeah, no, it could
but just saying it gives you very good information, but a different type of information.
That is very scary what you're saying here. So could you just give a little bit more detail? Let's say I had the absolutely spot on subtype identified cluster of cells in nuc-seq versus whole cell, and now I'm going to look at the counts per million, log counts per million, and between the two technologies. Who's up here? Who's down there? What's going on? You're saying that I'm going to have a whole lot that's off diagonal.
I think both techniques are fine
Is that right?
for
What you're saying?
to characterize the cells and to do the phenotyping, to describe the cell types and states. I think both techniques are fine, but they're not directly comparable. So they give you different types of information. So if you correlate information from one type or the other, it's not correlating as well as we would compare different techniques for full cell analysis.
But is that really that surprising when you think about you capturing such a small fraction of RNA anyways? You're just capturing a different small fraction of RNA.
Yeah, but I mean, what's the picture of the? We're trying to really understand the biology of that cell, and then how that cell relates to maybe others that are similarly differentiated. So now we've got this this view of that cell's transcriptome. It comes from nuc-seq versus whole cell with more mature mRNA. We've got to understand the information content in that expression vector in those two different technology reads, and so if we're getting stuff that is not particularly informative about the biology of that cell from the nuc-seq protocol, we need to know that, and so exactly what are we losing by going straight nuc-seq?
I think in the comparison,
I'm not seeing that big of a difference. I'm seeing tremendous enrichments deep down into nuc-seq, but I'm looking at nuc-seq data that's not undersampled 10x 3' labeled stuff. I'm just for fact separated nuclei with SmartSeq. Those are great signatures, but that are, I find highly comparable to the whole cell.
So from the comparison experiments that we've did, for example, we looked at neuroblastoma in several samples in metastatic breast cancer. We found the same cell types using both methods pretty much. And then we can use like Liger or random forest, and then the data sets align pretty well.
So we've had a, oh, go for it.
We have a first question from the live chat. So Josh Warburton is asking, has anyone had success capturing granulocytes?
Oh my God. I think we had this discussion yesterday in the immune section. Answer is no. So I can talk a little bit about personal experience. We captured granulocytes by accident when we did a really bad FICO. But in general, I think the challenge is that the granules prevent the RT reactions from happening. And so we actually don't get a lot of the RNA, even though we can get the cell surface proteins.
Maybe in Phantom, we were trying to profile primary mast cells, and we actually added heparinase to get rid of the heparin, because that was what was inhibiting the RT. Now, maybe you could actually add heparinase into the RT mix.
I think I need to make a comment. We got lots of granulocyte from the Michael Wells-Sig protocol. Sometimes I think the proportion is too much. Sorry?
So that's when you fix and...
No, we don't fix. These are all fresh cells.
And you still get granules? Oh, because you do well.
These are wells. I'm thinking about it could be because of droplet. Yep.
So we've seen better recovery of granulocytes in wells as well, and so I'm not sure if this is platform specific, but maybe this is a good time to talk about that. So I think most of us are probably doing some form of droplet-based single-cell RNA sequencing, maybe to complement well-based approaches, but I'll just open it up for other people who want to maybe chime in on their favorite platform that hasn't been discussed, or concerns about the current platforms that we're using. Everyone's doing 10x. Guoji, you want to take that one and share some of your experience on the microwells?
On which aspect? Sorry.
Maybe describe describe the method. I don't know for everybody.
Oh, okay. So we have these Argos microwells on this on the well. On this Argos microwell, there are around 100,000 well array, and then we just load the samples digested from fresh tissues, and then the cell will drop by gravity, and then we wash the plate. Then you will realize that 90% of the wells are empty, and then there are 10% that you have single cells. But if the dumplets, we just do a little bit of blowing, and then we put the beads inside. The beads will block the cells below, and then we do lysis. The beads capture the cells through the oligos linked to that bead. So this is, is that enough? Right.
And have you done, Holger, you've done some head-to-head comparisons of the various methods in terms of coverage. Was microwells or other well-based methods, or I guess high-throughput well-based methods, one of your comparators?
So unfortunately, we did not have microwell inside, but we did have SQL and isolate and one of different droplet-based methods, plate or microtiter plate-based methods. So we didn't see a systematic biases in terms of data towards one of the other protocols. So obviously, some are more scalable than others, but in terms of data quality, there didn't seem to be systematic differences between them.
So I think Guoji mentioned this earlier, and maybe not everybody is. I certainly don't see a University of Washington representative here, so I will mention sort of the split pool-based strategies, which they've really pioneered. The idea there is to use a single cell as your physical compartmentalization and do multiple rounds of combinatorial indexing. So it's going to be in situ reverse transcription followed by indexing with PCRs. And so you can imagine that doing in situ RT can be quite challenging because you need to semi-permeabilize the cells while keeping them intact. But in terms of scalability, and since now we're on skyRNA-seq 3, as far as I know, many of the efficiency issues have been significantly improved. We have seen some very promising data from that, and the scalability theoretically is into millions of cells per reaction. So, there's a question. Please use the mic and introduce yourself.
Hi, I'm Jeremy. I'm at Columbia. Someone was asking about direct comparison. So I think CZI funded Peter Sims do that for an HCA project, and the data should be online. I don't know if it's on the site, but they did a microwell versus chromium. There might be something else too. So there is a direct comparison out there that was funded. Peter Sims he's the one who created microwell-based RNA-seq. No, Columbia.
Do they do nuclei too, or?
I actually don't know if there's nuclei in there. They do it, but I don't know if it's in that data set. Yeah, but you could. I don't know if it's on the HCA website, or it should be on CZI though.
It's not on.
Okay. Yeah.
I guess maybe another thing that we should mention, this will transition us to the next point, which is when we're thinking about the different technologies and single nucleus attack it would be an outlier here for a second. Let's think about the RNA sequencing technologies. What are the parameters that all of you care about the most? Number of UMIs per sequencing depth, number of reads per cell. Yeah. Genes detected, I guess, is another common one, but I would love to hear all of your thoughts on that.
This is Bruce Arano. I just went through this list with Tim Tickle and who is it from the ingest pipeline. Anyway, just thinking about some general purpose metrics to have as annotations of a data set without going into cluster structure or anything like that. Just what would be useful to just get a over 220,000 foot view on a data set for a quick display preview on your shopping for data sets missions. And so the thing that I look for is the number of genes read per cell, the distribution of cells, what percentages of the total cells measured have, and so what's the drop-off rate look like? And I'd like to know that for total number of genes of genome, as well as then key subsets like translation, ribosome, and cell cycle, and then all the genes that are not in those kind of housekeeping non-tissue specific. So just get some kind of an overview of how much depth are we getting per cell, and the percentage of cells in the study that get to some level of gene count of unique gene types that are likely to contain interesting biology. Plus, then all the usual redundancy stuff and lack thereof.
Okay.
Holger, what do you have your list of things? What do you look for?
We tend to look at the usual suspects, so UMI, genes, but we tend to be very inclusive from the beginning. So we hardly do any quality filtering up front. So we look then at the distribution of populations and see how the cells that we would have filtered, how they look like in the context of the entire data set, and then we start filtering. So I think it's best not to, because it's very, very cell type specific, where to set the thresholds. And I think it's best to first include as much data as possible, and then do the quality control based on what you, how those bad quality cells or assumed bad quality cells, how they behave in the entire data structure.
This is something that I've noticed. Most of my work is in development and kind of cancer, where there's just a tendency to have more activated cells. And the thing that I'm seeing more and more adult, very mature, quiescent. Most adults are more quiescent. The there's probably likely very different distributions of genes, and that it's probably advantageous to be, to have a quiescent transcriptome, to be a mature stable cell, long-lived cell, and neurons are a very interesting contrast to that, and so I wonder what has anybody else seen that sort of a trend, and then what are the right kind of, are there different quality metrics that we need to think about for those, adult terminally differentiated cells?
Yeah, I mean, I think UMI definitely it's very important. I mean, it's a very important thing to consider, and also there are some cell types like T cells. I think it just express less transcripts than the other cells. But at current stage, no one can really define a bar because there are so many cell types, different tissues, and we have not even reached a complete agreement on technology, so it's hard to set a bar for that. But I think for this small chapter, I do think I need to comment on some of these technologies because it seems that not many people are technical here. So I think for HCA, it is quite important to to keep consistency of the technology, which I think 10 times genomics can be a good way to standardize different protocols. But if you really want me to comment on this different methods, there is one step that do not show up here is actually the amplification cycle during the sample preparation. And even for our micro-well-seq, initially we over-amplify, like we generate too much cDNA and only take a small fraction for that. And that's why, some of our initial experiment generates less genes and less UMIs. But now since we reduce the amplification cycle and take everything into transposase reaction, you just get more genes. And some of these, little tricks are hidden behind. And then for the split pool protocol, and one of the reasons I really like about this is actually they directly do transposase with unamplified cDNA, which means they only run one PCR cycle and then throw to sequencing. That truly reduces PCR cycles. But currently the technique is still under development, so we don't see this benefit. But once it gets out, so basically every transcript within the cell goes to sequencer without any amplification, you maintain the complexity of the data set. That's why I make the initial suggestion. But we still need to optimize a lot, like how to prohibit those transcripts from going out during the split protocol, how to preserve the RNA, and how to fix the cell, but not to block all these, poly A tails. But I think there will be a way.
Maybe back to your question. So we had a data set where we've expected a cell type, but we didn't see it in the final data set. No? We knew it should be there because then the FACS was clearly there. And at one point we figured out that those sets just express a lower number of genes. We had lower UMIs detected, and they just not fell into the distribution that were filtered out during quality control. So then we were loosening the threshold, and they were perfectly fine in the end. So it's just a case example of that filtering is important.
So I just want to comment on this a little bit. I think there's a few really interesting theoretical papers about how to think about how deeply to sequence. And one of my favorite ones is by David So, which is 1 read per cell per gene is optimal for single-cell RNA sequencing. It's in bioRxiv. I do think it's actually important to think about what the downstream effect, what the downstream analyses are going to be, whether or not you're going to cluster or you're going to do differential expression analysis, because the number of reads you sequence per cell is a power calculation. It should be about what you're looking for. So I think especially when we start generating data for this first phase and we're thinking about doing comparisons to disease individuals maybe later on, I encourage that all of us to at least be up to date on some of these theoretical methods that computational biologists and mathematicians have been sort of pontificating about for a while. And I'm sure many more of them will come forward. And related to that, I think another parameter that I don't know if any of you are really trying to optimize is cost. right? So we think about number of reads per cell, but it really is a cost consideration. And I don't recall, but I believe there's some papers published on this where if you're trying to optimize for cost, then what is the optimal experimental design? So I think that's also something that's worth considering. Of course, that's also sensitive to the particular technology that we're using, but suppose you use the same technology, you can still optimize for cost.
I mean, cost is an important parameter, right? The more cells you can do sometimes depends on the type of experiments you want to do. I said that cost is a very important parameter, and the lower the cost, the better for us. And that's one reason, for example, to do hashing. And as we are thinking more into the future, if we want to do we want to start looking into human genetics and do human genetic variation, we might need to do a much larger number of cells at the end of the day. So the lower the cost, the better.
So speaking of costs, there were some presentations at the HCA and also at the EMBO meeting earlier about the MGI sequencing. And I just want to ask any panelists here or any audience who have experience in MGI, and what are your thoughts about implementing this, or at least encourage Illumina and others to consider reducing the price?
Kirsten?
Yeah. So Sarah and Kedar from our lab gave the presentation. It was not a project I was closely involved with, but the upshot of it is that we had two identical sequence libraries which were sequenced through the Illumina platform and then sent to Beijing for sequencing. And the data looked pretty comparable, but it was cheaper. I think the cost was around 30 or 40% of the Illumina sequencing, something like that. But I don't want to go into more detail because I'm just not that familiar with it.
Hey, Joseph Powell here. I'll just comment on that. Our lab also did a comparison between NovaSeq, NextSeq, and the MGI Seq 2000 with 10x libraries of about 70,000 cells. And we had exactly the same pattern of essentially, well, there was a considerable difference in the performance between the V2 chem on the NextSeq compared to the MGI sequencer, but compared between the NovaSeq, it's essentially comparative performance. But we did see some interesting observations in the ability to call what I call sort of genetic architecture out of the library. So calling SNPs, and some of the libraries we used were CropSeq libraries. There were some interesting differences. The read length is different, so that obviously increases your ability to call SNPs. We saw an interesting observation in a much higher detection of the sgRNA guides from the MGI-seq compared to the NovaSeq. We're not entirely sure why this is. It's possibly due to differences in the library size. But overall, I mean, we were basically confident that it was similar level of performance. I won't cover.
Your sensitivity to detecting genetic variation is better.
I don't know if it's necessarily sensitivity. It's just we essentially sequence more of a transcriptome with longer reads.
Which you get with MGI.
Yes. Well, I mean, actually, even when we trimmed the reads, bioinformatically trimmed the reads to similar lengths, we still detected slightly more SNPs from the MGI-seq data. Just comment.
Maybe just to mention, so both papers are in bioRxiv. So if you
I think the Teichmann lab paper's just been published in Genome Biology. Ours is on bioRxiv. It's under review at the moment.
So maybe we can take the next sort of bullet point along with reducing batch effects, which is how should we as a community generate reference data, and should we think about doing duplicates or even more individuals of different genetic ancestries for reference human cell atlas? What is sort of the right number of individuals to profile before getting into so large scale that we're looking like a genetics community? Love to hear all of your thoughts, and I can talk about how, at least in my lab, we've been able to do some of this cost effectively.
I'd really like to hear the lessons from GTEx on that.
Well,
Why don't you start with the methods?
All right, fine. I'll start with the method. So, so I think the ability to sequence a lot of samples together has significant benefits, right? So, single cells basically are your compartments, and we could mix cells from many different samples for single cell sequencing. Most importantly, this reduces batch effects across samples. But the other thing is that it actually offers some cost advantages. So you reduce the dollar spent per sample. And so, of course, that data is all mixed and you can't deconvolve them unless you have a way to label your samples a priori. If you have genetically identical samples, there are some very nice methods for how to label them. So you could either use antibodies with a barcode to label them, or you could use these cholesterol molecules attached with a barcode that UCSF has developed to label your samples. What's nice about the cholesterol approach is that this will work with single nucleus as well as single cell sequencing. But the Broad has also developed, what's that? That's right. So the antibody approach does work also with single nucleus sequencing by tagging the nuclear pore proteins. Is that right? Right. So I think, so those are sort of experimental ways that you could increase the throughput. I should mention that Samantha Morris has a paper where you can introduce a virus into a cell, and that could be used as a hashtag. And so what my lab has developed, I think, is in some ways one of the easiest ways to multiplex is we just use natural genetic variation between individuals. And so here it requires that you multiplex cells or nuclei from individuals with different ancestry or different genetics. In fact, we could do first degree relatives, so not necessarily different ancestry, just different genetics. And what's nice is that experimentally, you don't have to do anything to label your cells. You just literally mix and go. The challenge with these approaches, and I think it's good that we talked about some of the sample handling considerations, is that you want to work quickly. Because if you have your cells or nuclei sitting around at room temperature, you're going to get all sorts of biases. So we multiplex up to 20 samples in less than 30 minutes. And we know that it works for single cell RNA sequencing, single nucleus RNA sequencing, ATAC-seq, and single nucleus RNA sequencing and ATAC-seq is data generated by others. So we actually didn't do this in my lab, but it works. And I think the key when we do these sorts of experiments is that if you look at least qualitatively at the t-SNE plots or UMAP plots, you have almost zero batch effects. You get the same data from each person because they were multiplexed together. And when you do have batch-to-batch variation, I think it also affords some, because that there's some anchors that you could use, you could potentially use better you could develop better batch correcting methods to fix any deviations in the batch. So has anybody actually tried these multiplexing approaches in the audience? Oh, great, cool.
Does anyone want to discuss them? I'll just briefly say that we're working with Jimmy and Arit to do some of these and on single nuclear preps for GTEx samples. Obviously, we'd love to go back to the GTEx study, which was a study of the effect of genetic variation on gene regulation, and take that to the cell level. So to the extent that we can, to go back to the resource that's there, we need to do nuclear approaches. And since we have genomes existing on those, we're using this approach where we demultiplex with genetics. And I guess, what are the downsides? Do we need to, in planning those experiments, how many individuals to pool? Do you need to sequence deeper, or what are the things you need to take into account?
So that's a great question. So again, we have this intuition. I think now there's some math that supports this. Is what you want to do is sample as many unique molecules as possible per dollar spent. Okay, doesn't matter how you get it. And so what these multiplexing approaches allow you to do is generate a more complex library. So yes, you get fewer reads per cell, but you actually get more unique molecules per dollar spent on the sequencer. And that's actually very useful for all of the downstream analysis, including clustering and differential expression analysis. And I should also add that if we are concerned about sort of, thinking about comparing the same approach across labs, for example, looking at different tissue cryopreservation methods, or thinking about heterogeneity between individuals, you could use multiplexing as a way to evaluate, right, these approaches by eliminating the fact that you're actually doing different batches that's correlated with the covariate they're interested in. So it's not just about throughput that way. So there's some other concerns with these methods, at least with the genotyping-based approach. You have to genotype the individuals a priori. And we think that's actually okay because you can just do a bulk RNA sequencing experiment, and that's actually nice data to have. We have a way to demultiplex without any prior information. But what happens is that you can't assign an individual back to a clinical covariate that you might be interested in, right? You still need some information to link them together. And often it's most cost-effective and quickest to just either do a genotyping array or a bulk RNA sequencing experiment.
So can you comment on doing that from bulk RNA versus whole exome sequencing?
Almost no difference. Yeah, at least in our experience.
But to get to the sort of GTEx level lessons about regulatory genetic variation, a lot of the SNPs that you want to know about aren't in the transcriptome. So you just really, the rich data information is going to come from
the genotyping, absolutely. Yeah, and so we have only a few minutes left, so maybe Joseph and we'll take any last questions. I'm happy to talk to other people about multiplexing.
Yeah, I'll just make two very quick comments to this. We do a ton of multiplexing using your demultiplex method. There's two things that are kind of interesting from it. One is, the SNP arrays cost, $40-50? They're less than the cost of storing a Guthrie card. So even aside from the use of that information for research questions, the use of it for sample mix-up deconvolution is really useful. I agree, priceless. But the other thing that's quite nice is because, and this has also been demonstrated by Jimmy's lab and others, is that because you can essentially detect the vast majority of doublets, particularly if you've got a high multiplexing strategy, you can then really push or superload high throughput systems. And so you sort of get double advantage of being able to reduce the cost of sample capture per individual as well.
So let me just comment on that for a second. And we started to notice this when we start to help people sequence tissue and single nuclei sequencing. The doublet detection is sensitive to the quality of your cells. So if you have a lot of cell death, you can imagine that if you have RNA from lots of different cells from different people in your mixture, then we tend to call higher doublets than we expect. But I think even calling what's a cell in those cases become more challenging. And we've definitely seen evidence of that. So it's important to really, everything that we talked about earlier is super important if we want to get really good high quality data out, making sure that we have high viability cells and nuclei.
Yeah, so in terms of doublet, then I would now advertise my micro-well seq-well as well. The well-based method has an advantage over, solving the doublet issue because you can always visualize the cell, before lysis. If the concentration is high, usually the local concentration defines the doublet rate, which means some regions or some droplet series have high concentration, then you will see doublet altogether. So if you see this kind of a region, you just blow a little bit or reduce the cell concentration or local concentration, then the doublet rate gets much lower. And also for the well-based, there are lots of washing steps. All these floating RNA and debris gets washed away. And then you have, less contamination, like what Jimmy has mentioned. So interesting. Different technology all has their own advantage.
You were commenting on reducing the cost per cell by multiplexing. Did you test the limits? How much you can overload a 10x lane before it gets clocked?
So it's interesting because we've done simulations. We can multiplex up to 128 people, but the challenge is really how do you handle that many samples without a robot? And so I would encourage all of you to go to Rahul Satij's website. We, two of us came up with a cost calculator to sort of figure out the optimal way to do your multiplexing experiments. Given the current doublet rates, it's about 25,000 cells per wells what we are able to get. Now these are PBMCs, so we've been sequencing. So for larger cells, I'm not sure if the same, I'm not sure what's going to happen to the system if you try to multiplex adipocytes if they don't explode, but they do explode most of the time. So I think we're in the last 5 minutes, and I want to make sure that we open the floor up to anybody who has some last minute questions.
So I guess for the sake of the community, will there be recommendations or standardized? There are protocols that are available, and there are in fact quite a lot. But question is, which one and how to standardize this so that we can actually benefit from integration of this data and protocol. So I know it's a very difficult question, and I don't expect it to be answered right now. But is there a plan by the STWG and others to suggest recommendations for platforms to tissue dissociations and so on?
I think we're getting out of this meeting that would be a very good thing for the STWG to do. That the protocols are very diverse. They're not all available. There's a lot of unique diversity, expertise. And to bring some of these together with that knowledge, even though we're obviously a community, it's a group effort, but to take a charge to do that, I think would be very valuable.
I totally agree, and I think as we said, we really started discussing that in depth, and we think it would be a really nice charge for the STWG moving forward, and something that we can work on in the next year. We've been working on it before, right? But I think it's something that we really need to invest a lot of time and thought of how to do it in a thoughtful and systematic way, I think.
Yeah, I think benchmarking is important, and partially it's done now for the RNA-seq methods. But I think the idea is to give guidelines, to give recommendations, but not really to pinpoint specific applications, because also diversity is good, as for tissue handling, but also for data production as well.
Yeah, I think guidelines is a good way, and suggestions, right? Yeah.
Thanks so much.
Thank you for joining us.
Thank you for everyone for your questions, and I'd like to thank one of the online audiences for asking that question as well.
And if you have questions in the future, then just email the STWG, and we're happy to answer.